Hello and welcome back to the Biochemistry for Health Sciences channel. Today's topic is starvation, the role of catecholamines, cortisol, and growth hormone. In the last two videos, 6.12 and 6.13, we discussed the secretion of glucagon and how glucagon affects the body during starvation. Several hormones either increase or decrease to regulate metabolism during starvation. So besides glucagon, as well as the decreased insulin levels that we see during starvation, there are many other hormones that also play a role in regulating metabolism during starvation, and examples that we'll discuss in this or future videos include ghrelin, cortisol, catecholamines, the thyroid hormones, as well as leptin. Before we talk about these hormones, here's a very important question that we need to appreciate in order to understand the effects of these hormones during starvation. And that is, what is the body trying to do during starvation, during this stressful situation that we call starvation? What's the body trying to do in order to survive? So remember, starvation means we are not eating, there is no food. Food, if you remember from previous videos, means fuel. So we don't have fuel. If you don't have fuel, then the body doesn't have enough energy. So the question is, what will the body do since it doesn't have enough energy? Because as we discussed in previous videos, energy is the most important thing that we need 24/7 in order to survive. So one of the things the body does is that during starvation is that it's going to reduce the usage of energy. Okay, and that's basically means it will try to decrease the basal metabolic rate, or BMR. So what is BMR? BMR is the minimum amount of energy that the body uses at rest in order to maintain essential functions for survival, such as breathing, circulation, as well as maintaining body temperature. And this reduction of BMR is carried out by thyroid hormones. So during starvation, the thyroid hormones will decrease in order to decrease BMR. Another thing the body will do during starvation is it will try to promote hunger so that we will find food and eat the food. Since ghrelin is a hormone that promotes hunger, this hormone will increase, while leptin is a hormone that suppresses appetite, so this hormone will decrease during starvation. Now, during starvation, there is no food, so which means no glucose is entering the bloodstream from our food. So this requires that the body starts making sure that there is enough glucose in the bloodstream, because the brain basically uses only glucose for its energy needs, as well as some other glucose-dependent cells such as red blood cells. So the body tries to make sure that our blood glucose concentration doesn't drop too much. It should be more than about 40-50 milligrams per deciliter, so that there is enough glucose to feed the brain as well as other glucose-dependent cells. So this means the body has to start making its own glucose or using glucose reserves in the form of glycogen, and that is predominantly done by the liver, So we see a lot of breakdown of glycogen as well as the synthesis of new glucose and also ketone bodies. We discussed this under starvation. One of the things we see in starvation is the formation of ketone bodies. So glucose as well as ketone bodies can then be used by the brain in order for us to survive. To make glucose by gluconeogenesis, we require building blocks, carbon skeletons, and these carbon skeletons come from the muscles. So we have hormones such as glucagon, cortisol, catecholamines that will cause the liver to increase glucose production. And we have hormones such as catecholamines and cortisol that will cause the muscle to provide carbon skeletons to the liver so that the liver can make new glucose by gluconeogenesis. So that's one of the effects we see in starvation is the not only the increase in glucagon, but also increase in these hormones, catecholamines as well as cortisol. Another thing the body will do is to make sure that this precious glucose that is being made by the liver is only used by cells that depend on glucose. So glucose-dependent cells, especially the brain cells and the red blood cells, and that this glucose is not used by other cells. So the body will try to stop the absorption of glucose by other cells. Okay, so adipocytes will not be able to take up this glucose. Muscle cells as well as other cells will not be able to take up this glucose. And this is called insulin resistance. Insulin resistance, which will prevent the uptake of glucose by other cells except the glucose-dependent cells. And one of the hormones that is very important for insulin resistance that we see increase during starvation is this hormone called growth hormone. So if the other cells, the non-glucose dependent cells, cannot take up glucose because of insulin resistance, they will need some other fuel, and that fuel would be fatty acids that come from triglycerides from the adipocytes. So we have these hormones, cortisol, growth hormone, as well as catecholamines. These hormones will cause triglycerides to be broken down into fatty acids, what we call lipolysis. And that fatty acids will not only be used by the adipocytes, but the fatty acids will enter the bloodstream and will be used by all the other cells, the non-glucose dependent cells as fuel. So this will now allow the glucose to be used by the brain, the red blood cells, as well as the other glucose-dependent cells, while the non-glucose-dependent cells, such as your adipocytes and muscles, will start using fatty acids as fuel during starvation. In this video, we shall focus on the role of catecholamines, cortisol, as well as growth hormone during starvation. Let's look at the catecholamines first. The two major catecholamines that we'll discuss are epinephrine, also called adrenaline, as well as norepinephrine, also called noradrenaline. Notice that epinephrine has a methyl group attached to the amine, while norepinephrine doesn't have the methyl group. Why are they called catecholamines? That's because this benzene ring with the two hydroxyl groups next to one another, in organic chemistry we call this substance catechol. So, and they also have amines. So that's why we call these catecholamines, because they have amines as well as the catechol structure. Most of these catecholamines come from the adrenal gland, the adrenal medulla part of the adrenal gland. In the adrenal medulla, we have the cells called chromaffin cells. And these are the cells that synthesize most of the catecholamines and secrete them. Norepinephrine, besides the chromaffin cells, do also come from the brainstem, from noradrenergic neurons. A few words about the synthesis of catecholamines in the chromaffin cells of the adrenal medulla. Catecholamines are made from the amino acid tyrosine. So this amino acid tyrosine is converted into DOPA, DOPA, and then DOPA is then converted into dopamine. So the DOPA is converted to dopamine, and then the dopamine is converted into norepinephrine. Then we have the enzyme phenylethanol-amine N-methyltransferase, or PNMT, and this enzyme catalyzes the methylation of this amine group in norepinephrine in order to form the methylated amine that we see in epinephrine. And this methyl group, the CH3 group, comes from the chemical called S-adenosylmethionine, or SAM. As we discussed in the previous video, SAM is a very important methylating agent that provides methyl groups to many chemicals that we find in our body. Once the norepinephrine and epinephrine are made, these catecholamines are then transported into vesicles for storage. So we have vesicles with the norepinephrine inside. And these vesicles also contain proteins that bind the catecholamines, and these proteins are called chromogranins. So during stressful situations such as starvation, we have these nerves, these sympathetic preganglionic nerves from the spinal cord. They release acetylcholine in the adrenal medulla. So we have acetylcholine released at the adrenal medulla, and the acetylcholine will bind these receptors, nicotinic acetylcholine receptors, on the chromaffin cell membrane. And when the acetylcholine binds these nicotinic receptors, the chromaffin cells become depolarized. And when they become depolarized, that will cause calcium to enter. So a lot of calcium will enter the cytoplasm of these cells. And as we discussed before, calcium stimulates exocytosis, and these vesicles will release catecholamines into the bloodstream. Okay, so the catecholamines are released into the bloodstream. Epinephrine forms the major catecholamine that is released in the bloodstream, about 80%, while the remaining 20% is norepinephrine. So that's how stress can cause the adrenal medulla to release the catecholamines into the bloodstream. So these catecholamines will, in the bloodstream, will go to different target tissues. So what are the major effects of catecholamines on the body during starvation? In the liver, as well as in the muscle, the catecholamines will stimulate the breakdown of glycogen. It will stimulate glycogenolysis. While in the adipocyte, the catecholamines will stimulate lipolysis, breakdown of triglycerides into fatty acids. So let's see how this happens. The catecholamines will bind receptors called beta-adrenergic receptors on the target tissue, whether it's muscle or liver or adipocytes. And once these receptors are bound by catecholamines, it stimulates a signal transduction pathway, which is very similar to glucagon. This receptor is also a G-coupled protein receptor. So this part here is very similar to what we talked about in glucagon, stimulation of adenyl cyclase, increase in the cyclic AMP, and that increase in cyclic AMP will then go and bind and allosterically activate this very important protein called protein kinase A, or PKA. So basically the catecholamines will bind to their target cells and activate PKA. In the liver and muscle, the PKA will phosphorylate glycogen synthase, making it from the active R form into the inactive T form. And when glycogen synthase becomes inactive, glycogen synthesis in the muscle as well as liver will stop. Now, in case of the enzyme that breaks down glycogen phosphorylase, that phosphorylation of glycogen phosphorylase is done by this protein called phosphorylase kinase. So phosphorylase kinase will phosphorylate glycogen phosphorylase from the inactive form into the active form. And once glycogen phosphorylase is active, that will increase glycogen breakdown. So how does PKA increase glycogen breakdown? Well, PKA will convert the inactive phosphorylase kinase into the active phosphorylated phosphorylase kinase. So PKA will phosphorylate this protein, making it active. And once this is active, this will now phosphorylate the glycogen phosphorylase, making the inactive form into the active form, and thereby increasing glycogen breakdown in the muscle as well as the liver. So PKA decreases glycogen synthesis, increases glycogen breakdown. So the catecholamines increases glycogen breakdown in the liver and muscle. Now let's see how the catecholamines increases triglyceride breakdown in the adipocytes, increases lipolysis. So one of the enzymes required for lipolysis is this enzyme called hormone sensitive lipase, or HSL. And this enzyme basically breaks down diglycerides into monoglycerides, and then these monoglycerides are further converted into free fatty acids and glycerol. So protein kinase A, one of the effects of protein kinase A in the adipocytes is that it will phosphorylate and activate this enzyme hormone sensitive lipase. And once it's activated, this enzyme moves from the cytoplasm to the lipid droplet, where it will start breaking down the diglycerides. Another effect of PKA is that it will phosphorylate a very important protein called perilipin 1, or PLIN1. Now, phosphorated PLIN1 is required for the activation of HSL. So that's another way by which this hormone-sensitive lipase will become activated, and that will increase the breakdown of diglycerides into monoglycerides, which then get converted to fatty acids and glycerol. Another enzyme that's required for lipolysis is this enzyme called adipose triglyceride lipase, or ATGL. Now this enzyme, in order to work, requires a cofactor and activator, CG158. So what happens is the phosphorylation of PLIN1 will decrease the binding of PLIN1 to this protein, this activator. This activator is now free, and the activator will bind ATGL. So in the presence of this cofactor, ATGL activity increases tremendously, almost 20-fold. And this increase in ATGL activity will cause the hydrolysis of triglycerides into diglycerides for lipolysis. PLIN1, when it's phosphorated, also does another thing. It increases the budding of lipid microvesicles, and that will increase the surface area to volume ratio. So instead of having one large droplet, we have many small droplets. And because these enzymes, these lipases, these are surface active enzymes, they sit on the surface and break down the lipid. So it's good to have a higher surface area. The higher the surface area, the more enzymes can sit on it and break down the lipids, the triglycerides into diglycerides, and then diglycerides to monoglycerides, and finally into fatty acids and glycerol. So that's how the catecholamines help the body during starvation. Increases the breakdown of glycogen in the liver as well as the muscles, and increases lipolysis in the adipocytes. Another hormone that increases during starvation is cortisol. So here's the structure of cortisol. Notice that this is a steroid hormone. This is a steroid hormone. So during stress, including starvation, it will cause the hypothalamus to release this hormone called corticotropin releasing hormone, or CRH. So the hypothalamus will release CRH. CRH will then cause the pituitary gland to release adrenocorticotropic hormone, or ACTH. Then the ACTH will travel in the bloodstream and will go to the adrenal glands, and the adrenal glands will then release the hormone cortisol. So that's how cortisol is secreted during starvation. So cortisol is synthesized in the zona fasciculata cells of the adrenal cortex. Basically, what happens is the ACTH that is released from the pituitary gland will come and bind to these cells, receptors. These are called melanocortin 2 receptors, which are found on the zona fasciculata cells, and that leads to a signal transduction, which causes or stimulates cortisol synthesis. Now, synthesis of cortisol is very similar to synthesis of the hormone aldosterone, which happens in the zona glomerulosa cells, except that these fasciculata cells do not have this enzyme, aldosterone synthase, and so instead of making aldosterone, they make cortisol. So once this cortisol is made, it's secreted into the bloodstream, and in the bloodstream, most of the cortisol is bound to serum protein, because remember, this is a steroid, it's not water soluble. Most of it is not water soluble, only a small amount. So 95% of it is bound to serum proteins, such as the protein corticosteroid binding globulin, or CBG, which is also known as transcortin. So most of the cortisol in the blood is bound to transcortin, there's always a little bit that's water soluble, about 5%, which is water soluble, and that would be the active free hormone that will carry out the biochemical or the physiological effects of cortisol. A few words about cortisol cell signaling. As mentioned earlier, most of the cortisol is bound to corticosteroid binding globulin or CPG. This is in equilibrium, in dynamic equilibrium, with the free cortisol. So the free cortisol, this free form that's not bound to the protein, will diffuse into the target cell. Because the membrane is hydrophobic and this is a steroid, so this protein, this hormone, this steroid hormone, cortisol, will diffuse into the cytoplasm of the target cell. So most of the receptors for cortisol, what we call glucocorticoid receptor or GR, most of the receptor for cortisol is found in the cytoplasm. So the cortisol in the cytoplasm will now bind the receptor, the glucocorticoid receptor. Now this receptor may have other proteins bound to it, such as HSP90. And so when the cortisol binds this complex, the HSP90 is removed, and now we have the glucocorticoid receptor bound to the cortisol. Now this complex of cortisol bound to its receptor will now enter the nucleus, and there it will bind DNA. So with the help of other proteins such as coactivators, this will now cause increased transcription. So this complex will bind DNA and cause changes in transcription. So various genes will be transcribed, mRNA will be produced, this mRNA will be sent out into the cytoplasm, and there the mRNA will be expressed into a protein. So cortisol basically increases the transcription of several proteins that are involved in proteolysis, in lipolysis, as well as in gluconeogenesis. So in the liver cells, cortisol will increase the amount of the gluconeogenesis enzymes such as PPCK, as well as glucose 6-phosphatase, and that will increase gluconeogenesis. In the muscle cell, cortisol will increase proteins of the ubiquitin proteasome pathway proteins, as well as the autophagy lysosomal pathway proteins, and increase in these proteins will cause muscle proteins to be broken down into amino acids, what we call proteolysis. So by increasing the amount of proteins, cortisol will increase gluconeogenesis in the liver and will increase protein breakdown or proteolysis in the muscle. Another thing cortisol does is that it will increase the breakdown of triglycerides in the adipocytes. Let's see how that happens. Cortisol increases the amount of ATGL as well as HSL, and these enzymes, when they're present in higher concentrations, will cause more rapid breakdown of triglycerides into diglycerides, and then diglycerides into monoglycerides, which eventually will be broken down into fatty acids and glycerol. So while the adipocyte is producing fatty acids by breaking triglycerides down, at the same time it is preventing fatty acid uptake from the bloodstream, from the triglycerides in the bloodstream. And these triglycerides during starvation would be mostly in the form of VLDL. One way this is carried out is by decreasing the activity of lipoprotein lipase. So without lipoprotein lipase, the triglycerides in the VLDL will not be broken down to fatty acids and glycerol, and therefore these fatty acids will not enter the adipocyte. And so this decrease in lipoprotein lipase activity is carried out by increasing the amount of this protein here called angiopoietin-like 4 or ANGPTL4. This ANGPTL4 is a very strong inhibitor of lipoprotein lipase. So the presence of ANGPTL4 will inhibit lipoprotein lipase and therefore prevent fatty acid absorption into the adipocyte from the bloodstream. So this means fatty acids from the stored triglycerides will be broken and sent out into the bloodstream, while uptake of fatty acids from the bloodstream will be stopped during starvation. Another hormone that increases during starvation is growth hormone. Growth hormone is secreted by the pituitary gland. There are cells in the pituitary gland called somatotroph cells. These cells synthesize and secrete a GH. The hormone GHRH, or growth hormone releasing hormone, which comes from the hypothalamus, this will stimulate the somatotrophs in synthesizing and secreting GH, while the hormone somatostatin, which also comes from the hypothalamus, will inhibit the somatotroph cells from synthesis and secretion of growth hormone. Growth hormone will then go to the liver and it will stimulate the secretion of this protein called insulin-like growth factor 1 or IGF-1. And IGF-1 has several effects. It increases tissue growth, it has insulin-like effects, and it's also a very important test for growth hormone disorders that we use clinically. Secretion of growth hormone from the pituitary is feedback inhibited by IGF-1, and there are also several other hormones that stimulate growth hormone secretion besides GHRH, and that includes ghrelin, which comes from the stomach, thyroid hormones, as well as the glucocorticoids such as cortisol. A few words about growth hormone signaling. Growth hormone will bind its receptor, GHR. This GHR is a class 1 cytokine receptor. Basically, it's a receptor that does not have intrinsic tyrosine kinase activity. So this is unlike the insulin receptor, which has intrinsic tyrosine kinase activity. So when the growth hormone binds its receptor GHR, that binding will cause an attraction of a protein called Janus kinase 2, which has tyrosine kinase activity. So this protein will come and bind to the GH-GHR complex, and then this protein, which is a tyrosine kinase, will now phosphorylate the cytoplasmic part of the receptor. So this cytoplasmic part will be phosphorylated. That means a lot of negative charges here. And this increase in negative charges at the cytoplasmic domain of this receptor is going to lead to the recruitment of several proteins. So many proteins will start getting attracted to this receptor. The major protein is called signal transducer and activator of transcription, or STAT. So we generally call this signaling pathway as the JAK-STAT pathway, JAK-STAT, Janus kinase STAT pathway. But besides STAT, there are also several other proteins that get attracted, including IRS, PKC, RS, MEK, ERK, and so on. And all of these, attraction of all of these proteins will then lead to various biological effects, such as the organization of the cytoskeletal part of the cell, the growth of the cell, as well as regulation of protein as well as lipid metabolism. So during childhood and adolescence, several nutrients such as proteins, minerals, vitamins will stimulate growth hormone secretion, and then this growth hormone will work synergistically with insulin as well as IGF-1, and it will stimulate growth, and the human being will eventually grow into an adult. During adulthood, when the final stature is reached, then growth hormone secretion occurs only during metabolic stress. And examples of metabolic stress include starvation, low blood glucose or hypoglycemia, exercise, as well as pregnancy. So in an adult, during the fat state, insulin will inhibit growth hormone. So growth hormone secretion is inhibited during the fat state. However, during starvation, which is a metabolic stress, growth hormone secretion will increase. So let's look at the role of growth hormone during starvation. So in starvation, the stomach is empty, and that causes the PD-1 cells in the stomach to synthesize and release this hormone called ghrelin. Ghrelin, very important hormone. A lot of research going on about this hormone. Ghrelin is known to have many, many different effects on the body. One of the effects is the effects on the hypothalamus as well as the pituitary gland. Ghrelin promotes hunger as well as increases the synthesis and secretion of GHRH. So this increased GHRH will go to the pituitary gland, and in the pituitary gland, ghrelin increases the receptors for GHRH as well as for ghrelin receptors, while decreases the somatostatin receptors. So this net result here would be increased synthesis and secretion of growth hormone from the pituitary gland. So the free growth hormone, the active growth hormone, will go to target tissues and cause various effects. Now, one interesting thing that we see during starvation is although growth hormone increases, there is no increase in IGF-1 secretion by the liver. This is called growth hormone resistance. So although GH under normal circumstances, GH increase should lead to a increase in IGF, but we don't see that in starvation. Now, there are two major effects that we know of growth hormone during starvation. One is insulin resistance, and the other is increasing lipolysis. So in insulin resistance, growth hormone actually blocks insulin signaling pathway inside the cell. Remember, even in starvation, there is a little bit of insulin. Insulin never goes down to zero. So there is insulin around, and growth hormone will block the effect of insulin. And this means that without any insulin effect, glucose uptake by the non-glucose dependent cells, muscles and adipocytes, will not happen. Okay, insulin resistance is also thought to be the reason why growth hormone increases glucose production from the liver, increases glycogenolysis as well as gluconeogenesis by the liver. So all of these effects may be due to the fact that growth hormone promotes insulin resistance. It blocks the effects of insulin. Let's look a little bit more on how growth hormone increases lipolysis in adipocytes. One of the effects of growth hormone that has been reported is that it stimulates this enzyme here, adipose triglyceride lipase ATGL, that breaks down triglycerides into diglycerides. And what seems to be happening is growth hormone through the MEK-ERK pathway causes the inactivation of this protein PPAR gamma, and that leads to decrease in the production of fat-specific protein 27 or FSP27. Now, FSP27 is a very strong inhibitor of ATGL. So without this protein, ATGL activity is not inhibited, and that increases lipolysis. Another way growth hormone increases lipolysis is through this pathway, PKC-ERK pathway, that leads to increased phosphorylation as well as activation of the hormone-sensitive lipase, as well as increased expression of this enzyme. So together by increasing these two enzymes, triglycerides will be broken down to fatty acids, and that will lead to increased lipolysis. Now, some researchers claim that the increased release of fatty acids into the bloodstream by the adipocytes may also be responsible for the insulin resistance that we see with growth hormone. Growth hormone also reduces the activity of lipoprotein lipase, probably through the inactivation of PPAR gamma. So this is the role of catecholamines, cortisol, as well as growth hormone during starvation. As mentioned before, starvation causes a drop in the thyroid hormones, especially T3, and this happens within 2 or 3 days of starvation. With reduction in thyroid hormone, the BMR will decrease, thereby conserving energy. The other two hormones, ghrelin, that promotes appetite, increases, while leptin, that promotes satiety or reduces appetite, decreases. In the next video, we shall discuss a little bit about these two hormones, ghrelin as well as leptin. So until the next video, once again, thank you very much for watching. Have a wonderful day, and we'll see you soon.